Lysosomes. Lysosomes are membrane-bounded vesicles produced by the Golgi apparatus. They have a very low pH and store powerful hydraulic digestive enzymes in an inactive state. Lysosomes assist in digesting material taken into the cell, and they destroy non-functional organelles and portions of cytoplasm. Materials can be brought into a cell by vesicle or vacuole formation at the plasma membrane. When a lysosome fuses with either, lysosomal enzymes are activated and digest the material into simpler subunits that then enter the cytoplasm. Some white blood cells defend the body by engulfing bacteria that are then enclosed within vacuoles. Within lysosomes fused with these vacuoles, the bacteria are digested. Sometimes a small amount of residue is left and then it is ejected from the cell at the plasma membrane. A number of human lysosomal storage diseases are due to a missing lysosomal enzyme. In Tay-Sachs disease, the missing enzyme digests a fatty substance that helps insulate nerve cells and increases their efficiency. The fatty substance accumulates in so many storage bodies that nerve cells die off. Affected individuals appear normal at birth but begin to develop neurological problems at 46 months of age. Eventually, the child suffers cerebral degeneration, slow paralysis, blindness, and loss of motor function. Children with Tay-Sachs disease live only about 3 to 4 years. In the future, it may be possible to provide the missing enzyme and in that way prevent lysosomal storage diseases.
4.5. The endomembrane system. The endomembrane system consists of nuclear envelope, the membranes of the endoplasmic reticulum, the Golgi apparatus, and several types of vesicles. The system compartmentalizes the cell so that particular enzymatic reactions are restricted to specific regions. The vesicles transport molecules from one part of the system to another. The endoplasmic reticulum. The endoplasmic reticulum, ER, consisting of complicated systems of membranous channels and saccules, flattened vesicles, is physically continuous with nuclear envelope. The ER consists of the rough ER, smooth ER, which have different structures of functions. Only rough ER is stubbed with ribosomes on the side of the membrane that faces the cytoplasm, and because of this rough ER has the capacity to produce proteins. Inside its lumen, rough ER contains enzymes that can add carbohydrate, sugar chains to proteins, and then these proteins are called glycoproteins. While in the ER, proteins fold and take on their final three-dimensional shape. Smooth ER, which is continuous with rough ER, does not have attached ribosomes. Certain organs contain an abundance of smooth ER and its functions depend on the organ. In some organs, smooth ER is associated with the production of lipids. For example, in the testes, smooth ER produces testosterone and steroid hormone. In the liver, smooth ER helps detoxify drugs. The smooth ER of the liver increases in quantity when a person consumes alcohol or takes barbiturates on a regular basis. Regardless of a difference in their functions, both rough and smooth ER from vesicles that transport molecules to other parts of the cell, notably the Golgi apparatus. The Golgi apparatus. The Golgi apparatus is named after Camillo Golgi, who discovered its presence in the cells in 1898. The Golgi apparatus typically consists of stacks of 3 or 20 slide-covered, flattened saccules whose appearance can be compared to a stack of pancakes. In animal cells, one side of the stack, the cis or interface, is directed toward the ER, and the other side of the stack, the trans or outer face, is directed toward the plasma membrane. Vesicles can frequently be seen at the edges of saccules. Protein-filled vesicles that bud from the rough ER and lipid-filled vesicles that bud from the smooth ER are received by the Golgi apparatus at its interface. Therefore, the apparatus alters their substance as they move through its saccules. For example, the Golgi apparatus contains enzymes that modify the carbohydrate chains first attached to proteins in the rough ER. It can change one sugar from another sugar. In some cases, the modified carbohydrate chain serves as a signal molecule that determines the protein's final destination in a cell. The Golgi apparatus sorts the modified molecules and packages them into vesicles that depart from the outer face. In animal cells, some of the vesicles are lysosomes, which are discussed next. Other vesicles may return to the ER or proceed to the plasma membrane, where they become part of the membrane as they discharge their contents during secretion. Secretion is termed exotosis because the substance exists in the cytoplasm.
Questions. Question 1. Contrast the structure and function of the rough ER with those of the smooth ER. The rough ER is stubbed with ribosomes on the side of the membrane that faces the cytoplasm. This allows the rough ER to make proteins. The smooth ER does not have attached ribosomes and its function depends on the organ, but most of the time it produces lipids. Number 2. Describe the relationship between the components of the endomembrane system. Proteins are produced in the rough ER and lipids in the smooth ER, and are carried and transported into vesicles, the Golgi apparatus, where they are further modified before being packaged in vesicles and then leave the Golgi. A summary of the whole thing: all the parts of the endomembrane system work together and process so that particular enzymatic reactions are resisted to specific regions.